Good morning, everyone. Sorry, good evening, everyone.
Good evening, sir.
Good evening, sir.
Good evening, sir.
तो पहली बात है कि हम लोग सुधरेंगे नहीं. You keep on doing nonsense chat in chat box. Why you are doing, I don't know. शादी करनी है बंगाल वाले से, वेस्ट बिहार वाले से, कर्नाटक वाले से. There already groups has been made. You can talk all those things in those groups. See now, as the chat box is off, now you are start raising hands. बस पूरा क्लास में यही करना है अब. So last class we started with transport across membrane. So anyone can tell. Okay, those who are new. We already finished 3 classes. This is 4th class. And since you got a link, you are eligible to attend this class. Okay. In between, I will open chat box. In that you can ask your doubt. Please, again, don't do all those nonsense. कौन कहाँ से क्या है. There are lots of time. 5 साल साथ में रहने वाले हो, पता चल जाएगा कौन कहाँ से. Okay. और बहुत ज़्यादा पढ़ाई कर ली तो 5 के 10 भी हो सकते साल. So don't worry about it. You get enough time to know each other. So can you tell me what we learned in last class? Anyone?
Sir, we have completed simple diffusion and facilitated diffusion,
Okay.
and even the differences between these two.
Differences. So anyone
Between
can tell
active
me
and
what's the
passive.
difference? Active and passive. Anyone can
Active
tell
and
me the,
facilitated, sorry.
yeah. Anyone can tell me the classification of transport mechanisms?
So divided in two bases. First one is in the basis of energy.
Hmm.
It has two examples. One is diffusion and the second one is active transport. In diffusion, there are two processes: in simple diffusion and facilitated diffusion.
Hmm.
Facilitated diffusion occurs through the help of protein channels that help that assist the transport. In active transport, there is a use of energy like ATP. Active transport is also of two types. First, simple and I'm forgetting a little bit.
So last class we finished classification of transport mechanism, why it is very important, and we already discussed passive transport, that is diffusion. In diffusion, both simple and facilitated diffusion and osmosis. So today we are going to talk about more about active transport. Active transport are the transport mechanism which utilizes energy either directly or indirectly from the ATP. So how we can, okay. So active transport are the mechanism which utilizes the energy either in the form of directly from the ATP or some other form indirectly from the ATP. And basis of the way they use energy, the active transport divided into primary active transport and secondary active transport. These are the transport mechanisms which use protein, carrier protein or transport protein as their channel to transfer the substances from one side to the other side of the membrane. Apart from this, another one more mechanism of active transport is another mechanism that doesn't involve a protein channel, but it goes along with the whole membrane. So it transfers through the part of the membrane by forming vesicle. So they are called basically vesicular transport mechanism. Vesicular transport mechanism also utilizes lots of energy. That's why it also come under active transport. And this vesicular transport mechanism for larger substances like protein molecules or even microorganisms or sometimes fragment of the cells or sometimes complete cell. So that mechanism is called endocytosis and exocytosis. So end of this class we'll talk about that endocytosis and exocytosis also. So when we are using energy, we'll use energy invest that energy to work against the flow. So this active transport always work against a gradient. Either maybe it is a concentration gradient, we call it chemical gradient, or it can be electrical gradient. So it always bring substance from low concentration to the high concentration. It is like it is going from water is moving from the well to the water tank. So whenever we are want to put water from well to the water tank on high up, we use energy either in the form of electrical energy or we can use diesel pump. So in any form, we are spending energy so that it will move from water to the higher tank. So this same way here also we use energy, and that's why this moment is also called as a uphill moment. So from lower portion, sorry, from lower portion it goes somewhere higher. So moment is upward direction, and that's why we are called it as a uphill moment. So substance which can transported by this active transport, here we are talking about non-vesicular, which going through the membrane proteins, that only. We are not talking about endocytosis and exocytosis. So what are the various substances which can be transported by this active mechanism? Most commonly and very utilizes ions like sodium, potassium, calcium, chloride, magnesium, iodine. All these ions are can be transported against the concentration gradient, copper, uh, iron. They are moving against the concentration gradient and uh, concentrated at the one particular place at a much higher folder than other side. Second thing, there are some ionic substances also which can be transported by this mechanism, glucose, amino acids, and urea. So they are also moves against concentration gradient. So depend upon how Josh, can you switch on your camera? Just Badoria, I guess. Badoria. Bodaria. Yeah, Aditya, it's good. Disable annotation, disable all those things, traffic rules लगा लो, signal लगा लो. If you people don't want to follow the rules, then what is the use of doing all those things? These all facilities is given so that we can communicate. Even in this corona period, we can do more productive things. This is not like, okay, we'll do this, we'll do that, and we'll try to control. But if your intention is not to learn, then what is use? Now you raise hand, I make you panelist, and now you are doing all those things. किसी को कोई मैसेज देना है? जश? प्रियंका, are you new?
Yes, sir.
Okay, so this classes has been already started. I recorded all these classes, so the recorded classes is available on YouTube. You message me, I will send you the link. Okay?
Okay, sir.
And if you have any queries, any doubts, you can ask me. I will explain uh, those doubts and try to clear it. Okay?
Yes,
Anyone
sir.
who want, they can message me. Okay?
Hello, good evening, sir.
Good evening.
Sir, I want to join the WhatsApp group Prosium so I can follow up.
Actually, I'm going to leave that WhatsApp group and I will keep only one Telegram group. Okay, you WhatsApp me, I will send you the link of Telegram group. Okay? The WhatsApp group is already filled. 257 members is already there. So we can't add, so I shifted to Telegram group so that all student can be communicate in one group. Okay?
Okay, sir.
Sir, I'm also new. So how can I message you?
सबको पर्सनली बताना पड़ेगा सब. व्हाट्सएप करो. Write down my number, those who are new. Plus 99 plus 996 7700 39261. हो गया?
Please repeat.
Plus 996 7700 39261.
Okay, sir. Let's continue class, sir.
Sorry?
Let's continue
He's
class,
saying to
sir.
continue.
Don't message right now. Message me after class. So there are two transport mechanisms, primary active and secondary active. So first we'll talk about primary active transport. This primary active transport are the transport which utilizes the energy directly by the breakdown of the ATP and use that energy to transport the substances against the concentration gradient across the cell membrane. Okay, so here they directly split the ATP into ADP and inorganic phosphate. Yes, गिरीश चौधरी, तुम्हें 4 बार मैं प्रमोट कर चुका हूँ, अब नहीं हो रहे हो तो मुझे नहीं पता. ओके, यही करते रहते. These are the substance transport mechanism. Since they split the ATP into ADP, and that will release the huge amount of the energy, and that energy is utilized. So, since the split, whenever any chemical or protein will use to split the substance, we call it as a use the term ACE. Okay. Example, lipase. लिपिड को स्प्लिट करने वाला लाइपेज. Similarly, we use this ASE prefix suffix to the that particular substance. And since this pump this substance mechanism split the ATP, we use word ATPase. Okay, so many times primary active transport also utilizes with the name of ATPase, like sodium potassium ATPase. Okay, since they lyse the ATP into ADP and inorganic phosphate. Another term we use is pump, because just like a motor pump, or it pushes the substance from low concentration to high concentration with the help of energy. This is also called as a pump. So sometimes we use like sodium potassium pump or calcium pump or potassium hydrogen pump. Like that we use sodium hydrogen pump we use. So we use another word is pump. So sodium potassium transport substances, or sodium potassium transporter, sodium potassium pump, or sodium potassium ATPase. Like that we can give different nomenclature to the primary active transport. Okay. Out of these are just 2-3 examples. There are many examples of primary active transport. These are which commonly found. Out of these, sodium potassium pump is one of the commonest pump, and which is seen in each and every living cell. मेरा वॉल्यूम तो बड़ा है, एक मिनट. Is it okay now?
So it's echoing,
Yes,
sir.
sir.
Can you hear
No,
me?
sir. It's common. Yes, sir. Normal.
Yes, sir.
Which is better? Now I'm changing
Yeah,
again.
now it is better, sir.
Now,
No,
previous one.
it is okay, sir.
So both are good.
Okay, then I'll keep this. Tushar, can you hear now?
Yes, sir.
So, sodium-potassium pump is which commonly seen in almost all cells. So it's very easy to extract from the cell, and it is the one of the pump which has been highly studied. So whenever we are describe the uh, primary active transport, where we usually talk about sodium-potassium pump. Okay, so we'll describe here primary active transport as a sodium-potassium pump. As I said, it is already present in all the living cells in our body. It transports the sodium ions outside the cell. From inside the cell, it moves the sodium ion outside the cell. So consider this is a cell. So it moves sodium ion outside the cell. So sodium ions will move outside the cell membrane, while potassium ion moves inside the cell. Okay. Now, if you remember my first slide of the transport mechanism, first slide was transport mechanism. I described that there is an extracellular fluid and intracellular fluid. Extracellular fluid, sodium concentration is higher, while in intracellular fluid, potassium concentration is higher. So movement is from low concentration it is going towards the high concentration. From low concentration, it is going towards the high concentration. And this mechanism is responsible for such a vast difference of sodium and potassium concentration across the cell membrane. जो extracellular fluid or intracellular fluid में difference है of the sodium and potassium concentration, it is because of the sodium potassium pump. Another thing,
Sir, is sodium attracts the water in the cell? If sodium nodes moves out from the cell, it attracts the water inside the cell and it increases the turgidity of the cell. Sir, it is right.
yeah.
So sir, that the cell is sir burst. So sir, it is necessary to move out from the cell.
Hmm.
Okay, sir.
Okay, so sodium is moving out and potassium is moving in. Apart from this, the 3 sodium ion moves outside and only 2 potassium ion moves inside. Okay, and that create the electrical imbalance across the cell membrane. So हर बार एक positive charge cell से बाहर जाता है. Whenever the sodium potassium pump activate, it will make a difference of 1 positive charge less inside the cell, and that's why this sodium potassium pump is also little responsible for electrical potential difference across the membrane. Okay, so when we are talking about membrane potential, we'll talk about this. This is not solely responsible, but it is also one of the factor for the sodium potassium. It is factor for the membrane potential. Okay, now we'll see the structure, how the sodium potassium pump look like. It is made up of 2 separate polypeptide. That is a larger subunit alpha, which has somewhere molecular weight around 1 lakh, and smaller subunit beta, which has a molecular weight somewhere around 55,000. It present across the cell membrane, so it almost moves throughout the cell. Actually, इतना बड़ा नहीं होता. It just to demonstrate you, to show this side. So it is somewhere just above the cell membrane, but it is throughout the cell membrane. So it exposed to the extracellular fluid also, and it also exposed to the intracellular fluid also. So it is throughout the membrane. Out of these, alpha unit is main unit, while beta unit is a supporting unit. So mainly alpha unit, which is usually concerned about the movement and all those things. Beta usually act as an anchoring of the unit. Okay. If you see this structure detail about, so we'll see the how this alpha subunit has a the alpha subunit and beta subunit has a inbuilt total ATPase activity. So here they had a enzymatic activity which splits the ATP into ADP and inorganic phosphate. So another side, alpha subunit has inside the cell. This is extracellular fluid outside, and this is inside. Okay. Another site is the binding site for the ATP. ATP bind to the alpha subunit. ये ध्यान में रखना छोटे छोटे points है, and you'll get MCQs on this point. Okay. So alpha subunit has a binding site for the ATP binding site. Another that there is a phosphorylation site where the phosphorylation of the ADP occur. Whenever the ADP is there attached, it can occur. And then there also has a binding site for a 3 sodium ions inside the cell. So inside the cell, it has a binding site for the 3 sodium ion. Now you come to the outside. In exterior, alpha unit only has a active site for the potassium ion. So 2 potassium ion binds to this binding site which present on a alpha unit. Apart from this, there is Aubin chemical which can block the activity of the sodium potassium pump. That Aubin ka binding site be present on the alpha sub unit. Okay. Now how it works? Did you understand the structure?
Yes, sir.
Okay.
Okay. So what is meaning of ECF and ICF?
ECF extracellular fluid means this is the exterior of the cell, and ICF intracellular fluid, this is interior of the cell. So what is the structure? The structure is inside the cell, and this structure is outside the cell. Understand? This is cell membrane. ये जो lipid
So
bilayer दिखा रहे हैं ना,
once again
this
repeat.
is sorry.
Once again repeat, sir. Repeat.
ECF is extracellular fluid. So this portion is okay. 1 minute. So this is the cell. Okay, and here is this transport protein is there. हमने इसका magnification इधर किया है. So this outside the cell, this is ECF, which is outside, and this is ICF, intracellular fluid. Okay. So this structure is showing which is inside in this portion, this structure, and this structure is showing which is outside the cell. Now understand?
Yes, sir.
Okay.
Thank you.
So now we'll see how the sodium potassium pump work. So what happened? First, there is ATP which bind on the active binding site of the alpha subunit, and that activate the this whole transport. After that, whenever there is a sodium concentration inside the cell increase, because of this has a high binding site, sodium ions start attaching to this binding site. And once all 3 sodium ions attach, this ATP will bind here, and now this will both activate the ATPase portion of the pump, and this ATPase will catalyze the ATP into ADP. Please mute your mic. ATP is catalyzed from ATP is catalyzed to ADP plus inorganic phosphate, and also release the high energy. Okay, these phosphates bonds are called high energy bonds. Okay, third bond is a much has a strong energy, and ATP also work as an energy currency in our living cell. So that high energy will be form there, and it charge this whole mechanism. It is a pump. Now what happen at the same time? What will happen once this is sodium chain ion ATPs will be there charged. Now this portion will increase the affinity for the potassium, and potassium ion binds here. Once potassium ion bind here, this will make now conformational changes. Conformational changes means structural changes. Now how we can see? Okay, now this is, suppose consider this is the protein molecule and this is its structure. Now when high energy bond will conformational changes, it will change its structure. And now it arrange separate orientation. By making these conformational changes, this portion moves inside. The ATP binding site moves inside, while sodium binding sites moves outside. So 2 potassium ion moves inside and sodium binding site moves outside. Moment this sodium binding site moves outside, this alpha unit loses the affinity for the sodium, and sodium will release into the extracellular fluid. As it loses the affinity for the sodium, at the same time it loses the affinity for the potassium also, and potassium ion also moves inside the cell and release inside the cell. And once this both will be released, it recoil and it get back to its normal position. Moment it comes and get back to its normal position, and now here again the ADP will move and ATP will attach to this portion, and it will again come to its original state. Again, if sodium ion concentration increase, again it will bind it and activate the same cycle, and it will keep on pumping the more sodium ion outside and more potassium ion inside. Mechanism समझ में आया?
Yes, sir.
Yes,
sir.
Yes,
Yes, sir.
So I am opening the chat box. So if you want, please ask. You can ask me question. Obin is a chemical which can block this pump. Dephosphorylation means it removes the one phosphate molecule from the compound. That's why we call it dephosphorylation. ATPase ATP को split करने वाला ATPase.
Sir, your voice is lagging.
No, sir, your voice is not like
No, sir.
maybe.
Conformational changes. Suppose some protein is like this. Now, whenever energy is utilized, it will make some shape. Another shape will be changed. That is called conformational change. Obane is alkaloid, and that can block the sodium potassium pump. Function of B side. B side is act as an anchor and hold the whole alpha unit at that position. Uphill movement is done by the active transport, while downhill movement is done by the passive transport. By this mechanism, only sodium and potassium balance is restored. We don't want equal concentration of the sodium in both intracellular and extracellular. No, this is not depolarization. This is normal process occur throughout our body. We don't need ADP, we need ATP. Whenever ATP is converted into ADP, it will release the bond energy. Then again, this ADP goes to the mitochondria, and in mitochondria, again this ADP is converted into ATP with the help of electron transport mechanism. Both ionic transport occur through both active and passive. Yeah, it is a resting state. सारा एक ही दिन में सीखना. व्हाट अबाउट अदर आयंस पे आऊंगा ना, एक एक चीज सिखाऊंगा. It occurs in almost each and every cells of our body. Yeah, alpha subunit has a ATP binding site. अभी बताया beta cell is just an anchoring cell. Yeah, you know RBC also. What is last point?
Deep oscillation of the substance which returns to its provided confirmation. Sir. Last point.
Okay. Okay, good. Okay. Now first thing, restoration means it's recoil. किसी एक पोजीशन में आ गया एनर्जी में, जैसे इलास्टिक को स्ट्रेच करते हैं, so we need energy to make the change of the shape. The moment you release, it again come back to its original position. That is how it restore its original position or recoil to its original size and shape once the energy has been removed from the that portion. Okay. Another point what you ask. Yeah. Last one, phosphorylation. When phosphate bond is added to the any substance, we call it a phosphorylation. So this causes phosphorylation of ADP and convert it into again ATP by utilizing the energy.
So Telugu understanding?
Sorry?
Telugu.
No. तेलुगु में MBBS करना है?
नहीं sir.
ठीक है. Okay, before going to the functions of the sodium potassium pump, we need to talk about factors which regulate the sodium potassium pump. Now you will ask, sir, कहाँ पे होता है mechanism, क्यों होता है, कितना होता है, क्या होता है? This sodium potassium pump continuously acting in our body, and it keep on pushing sodium ion outside the cell and putting potassium ion inside the cell. If you want to know how much amount this pump is used, so almost 30% of your total BMR energy expenditure. You know what is BMR?
Basal metabolic
Basic metabolic
Basic
rate.
metabolic rate.
Metabolic
This is the
reaction.
amount. Yeah, this is the amount of the energy. अरुण, तुम बोल के बता सकते हो क्या बताना चाहते हो? Maybe it's panelist or participants take last 15 minutes doubt session. I don't get you.
सर डाउट सेक्शन अगर 15 मिनट्स बोल सकते हो सर, डाउट्स निकलने के बाद लास्ट 15 मिनट्स को पूछता हूँ ना सर, एक
Okay,
टॉपिक को बीच में पूछने का, लेकिन
you don't want
इधर
it
वो
in between. You don't want it in between. You want by the end of the session. Is it so?
हाँ एकदम पूरा हो जाता ना सर डाउट सेक्शन, तो डाउट क्या क्या एकदम पूरा हो जाता है थोड़े बार इसको
Actually,
हमारे जैसे तुम मैं पैनलिस्ट हूँ, दूसरे बच्चे आ रहे मैं पैनलिस्ट बात तो मैं भी पूछ सकता हूँ, तो उनको भी लगता ना डाउट
I could not able to hear
पूछना
you.
तो
I
एक
could
बार
not able
बोलते
to
हो टाइम
understand.
से बोला
I could not able to understand. Your sound is lagging, muffling sound.
सर लास्ट 15 मिनट्स डाउट सेक्शन
अभी बीच में डाउट्स नहीं करने, सिर्फ आखिर में 15 मिनट्स में करना है.
नमस्कार,
Yes.
ऐसे कंटिन्यू कर सकते हैं.
Let him finish, na. Let Arun finish first. Yes, Arun.
सर डाउट्स सेक्शन पूछने का कौन था लास्ट 15 मिनट
लास्ट मतलब बीच में नहीं चाहिए आपको, लास्ट में चाहिए, ऐसा आपका कहना है?
सर बेटर रहता अब पूरा डाउट्स एक एक बार हो जा सकते, थोड़े थोड़े बार बोल तो क्लास को पता
Now see,
एकदम पूरा हो जाता,
your
क्लास हो जाता
yeah,
सेपरेट, ये भी हो जाता ना वैसे बोल.
it's good suggestion. Now you see, I already explained 3 times about the organ binding site, and we are very particular about thing which is not important at all, हमें उसमें ज़्यादा इंटरेस्ट होता है. Now I'm talking about more about how the transport is occurring and all those things. The organ is just to show that there are some chemical which can block this activity. And I already 3 times explained. Still there is a question is coming. Sir, what about organ binding site? What about organ binding
इसीलिए बोल
site?
रहा हूँ सर लास्ट 15
It
मिनट्स
is
हो जाता
just
तो पूरा हो जाता है
alkaloid.
एक बार.
Yeah. लास्ट में बोलूंगा, लास्ट में तो और भी हो जाएगा. ठीक है, अभी लास्ट में करेंगे. This organ is a chemical alkaloid. Alkaloid are the chemical which taken out from the plants. Okay, we can call it the poisons also. Okay, so those alkaloids, when given to the any individual, they can block the sodium-potassium pump. So how they block? They bind by binding this site. Okay, they stop the activity of this pump. Now you got everyone clear about organ binding site?
Yes,
Yes,
sir.
sir.
Okay.
Sir.
Yes.
Yes, sir.
Okay, now there are more important. I'm talking the this pump is so high concentration present in our body and they are so much active that a 30% of our total energy expenditure, the basic metabolism में जो minimum required for survival of the body, energy we spend in that energy expenditure, 30% energy spent only to activate the sodium potassium pump. To keep the sodium potassium pump active, only for that purpose, is 30% of the energy is utilized. It is such an important pump in our body, and each and every cell it is present. Okay, so this is important. Now, how this pump has been regulated? As we already talked about, this Obin can block. There another drug called digoxin, which can also block this pump. But this is about the more of pharmacological part. Physiologically, how it regulated? It regulated by the only one factor, and that is presence of the sodium ion inside the cell. More is the sodium ion inside the cell, more this pump will become more and more active, and it will keep on removing the sodium from the cell and pump it outside the cell or move it into the extracellular fluid. Less is the sodium ion, less will be the pump. Even the sodium ion has such a high effect on this, it can increase the numbers of the pump. So it causes upregulation and downregulation also of this pump. So only regulatory factor is sodium ion. Moment sodium ion increase, the pump will become more active. Okay, now what is the function of the sodium potassium pump? First and most important, आप में से किसी एक ने बताया था, it controls the volume of the cell. Now first understand the behavior of the sodium. So sodium is more of the hygroscopic. So hydroscopic. So it always carries the water along with it. So whenever the sodium moves inside the cell, along with the sodium also has its own friend, that is chloride. Chloride will also follow along with the sodium. So chloride will also come inside the cell. Sodium chloride will have hydroscopic action, and it will pull the water inside the cell. And this is how it will start increasing the size of the cell. And one point will come when cell membrane will not able to hold that much volume of the cell, and it will burst, and that causes the lysis of the cell. So if sodium potassium pump doesn't work in our body, it can cause death of the cell by just simply causing lysis of the cell. Now someone will ask question, if sodium is moving outside, if it is not coming inside, then even if sodium potassium pump is not work, then also cell volume will remain? No, this doesn't happen. The sodium will keep on leaking inside the cell throughout the cell lifetime, because of very high concentration of sodium outside and very less concentration of the sodium inside. So it will keep on taking leaking inside the cell, and that what leaking sodium inside the cell has to pump outside. So it has to continuous work, and it keep on pumping the sodium outside the cell. So this is one of the important function of sodium potassium pump. Then come to the electrogenic activity. किसी ने सवाल पूछा था सर इलेक्ट्रिक पोटेंशियल के बारे में बताओ कैसे. तो as we already know, sodium potassium pump moves 3 sodium ion outside the cell and 2 potassium ion inside the cell. They will be causing net loss of 1 positive charge outside the cell. So cell के अंदर से cell के बाहर चला जाता है, and that will create the electric difference across the cell. So this is not the only factor which is responsible, but apart from other factors also responsible. So when we are going to talk about the resting membrane potential, we'll talk about the other factors which are responsible for the membrane potential. Now, what is the importance of this generation of the membrane potential? Membrane potential is very important to work the nerves and muscle properly. If now membrane potential is not there, nerves and muscles will not work properly. They work by changing the membrane potential and now transmit the signal via action potential. And similarly, muscles receives via signal via action potential, and also muscle contraction also occur via action potential. Not only this, but also release of the various chemicals, release of the various hormones are also occur through these electrical potentials. So that electric genic activity is very important. Then now come to the other pumps. So another pump which is usually asked is calcium pump. This calcium pump usually work as a solo. It doesn't like a sodium potassium work together. This calcium pump usually works solo. High number of calcium pump you will see in permeability is related to diffusion or passive transport mechanism. So sodium potassium pump doesn't have much effect on the permeability of cell membrane. They pump the chemicals against the concentration gradient. Okay. Now come to the calcium pump. These calcium pumps are more seen in concentration in skeletal muscle. Those channels are called Raynaudin channels, and they pump the calcium against the concentration gradient from inside the cytoplasm into the sarcoplasmic reticulum, and that can be as high as a 2000 times. 2000 times to 20,000 times higher concentration of the calcium inside the sarcoplasm as compared to in the cytoplasm. Okay, so sarcoplasmic reticulum may much higher concentration will be there with the help of calcium pump. Okay, and this is how this calcium pumps keep on pushing the calcium outside. If you see the calcium pump also present on the cell membrane, and those cell membrane will keep on pushing the calcium outside the cytoplasm, and the concentration difference between intracellular fluid and extracellular fluid of the calcium is 1:1000 times. Okay. Another important pump which many times we talked about is potassium hydrogen pump. Since hydrogen plus ion is involved, many times hydrogen plus is also called as a proton, because it is just single proton and there is no electron. So that's why this pump is also many times referred as a proton pump. And this potassium hydrogen pump is seen in parietal cells of the stomach. In parietal cell of the stomach, this proton pump or potassium hydrogen pump, here the potassium moves inside the cell and hydrogen pump into the canalicula of the parietal cells. And with another mechanism, chloride will be added into the canalicula, and that will help to release the hydrochloric acid inside the stomach. Similar pump will be seen in the kidney, but that is sodium hydrogen pump, where the sodium ion is exchanged with the hydrogen ion and causes acidification of the urine. Okay, This potassium hydrogen pump, we use a very common drug which can block the activity of the potassium hydrogen pump, and these are called proton pump inhibitor. You might have taken it very regularly, omeprazole, pantoprazole, or rabeprazole, tretinoin, PAN 40, PAN D,
Sir, for
all these
acidity.
medicine, for acidity, so that it can reduce the acid secretion in the stomach. Okay, so these are the few examples of primary active transport, and among these only it will ask any questions, any doubt regarding primary active transport. Now come to the secondary active transport. Now, when we are coming to the secondary active transport, there is lots of confusion in various books. So many books has been given like one facilitated diffusion. There also they are given a mechanism like a co-transport or counter-transport, all those type of the mechanism that is not a part of facilitated diffusion. Facilitated diffusion is just simple facilitated diffusion across the channels. So when there is a co-transport, counter-transport, symport or antiport will come, or sometimes uniport will also be there. So these are all related to secondary active transport. Here in secondary active transport, energy is not directly utilized from the split of the ATP. So ATP से directly energy नहीं लेते. So it is not, there is no dephosphorylation of the ATP, and it will not convert into ADP and inorganic phosphate and all those things. But the energy is utilized by some other way, and that leads to formation of secondary active transport. So usually this transport mechanism occur when they are coupled with some other chemical. So most common chemical which coupled is sodium. So we can call it a sodium co-transport or sodium counter-transport and all those things. And along with the sodium, this chemical will be transported. So now what happened? Suppose this is a cell and one chemical has to move from outside to inside the cell. So what happened? The particular chemical, okay, we'll say A. A will attach to this transport mechanism. Another binding site will be for the sodium. So sodium will attach to this transport mechanism. Now concentration of the A is much higher inside the cell. आपके यहाँ पे ये बहुत ज़्यादा है यहाँ पे, is much higher. But sodium concentration is very less. There hardly sodium concentration. But the outside sodium concentration is much higher. Now because of this high concentration of sodium outside and very less concentration of sodium inside, there is a concentration drag from outside to the inside, and along with the drag sodium will move inside. So when sodium moves inside, it holds the hand of this chemical, and it also pulls the chemical inside the cell membrane. The moment it goes inside the cell membrane, sodium will release into the cell. Okay, so यहाँ पे और जैसे सोडियम रिलीज होगा, this receptor protein, channel protein also loses affinity for the A, and A is also released into the cell. And moment this both effect will occur, this receptor again come back to its original position. Again, what will happen? Again, sodium ion attach, the chemical attach, again sodium will move inside, and that will keep on concentrating the A inside the cell. Okay, this is what called co-transport or symport. In same direction, both chemicals are moving. Now opposite. Now suppose the chemical has to move outside, then against the concentration gradient there will be opposite side. यहाँ पे attach हो जाएगा और इधर sodium attach होगा. Sodium moves inside. This will pull outside. So this is how the co-transport and counter-transport work. So here we have this some example is glucose transporter, which you can see in intestine when we are absorbing, and also seen in various cells where the glucose inside the cell is required, and also seen in a renal tubule whenever the reabsorption of the glucose molecule has to be occur. Okay, so this is an example of symport, and it can be clubbed with other chemical also, but sodium is one of the common chemical. Now here if you see, This chemical has a binding site. One has a binding site for the sodium, another has a binding site for the calcium. Glucose. Now, as the sodium concentration much higher in the extracellular fluid, sodium will come and attach to the binding site. Moment this attach, another binding site for the glucose will increase the affinity for the glucose, and here glucose molecule will also attach. Now, when both will attach, this whole transport molecule will become active and it will causes conformational changes, and this is called beveled moment. वो सीसॉ जैसा मोमेंट होता है ना, तो सीसॉ टाइप ऑफ मोमेंट, जो ऊपर है वो नीचे आ गया, जो नीचे है वो ऊपर गया. So seesaw type of the moment or beveled moment occur, and along the concentration gradient, sodium moves inside the cell. And since sodium moves inside the cell, it also pulls this portion inside the cell, and both will comes inside. As it comes inside, sodium concentration is very less inside the cell, so sodium will release from this side. And as the sodium moves, this site also loses the affinity for the glucose, and glucose will also moves into the cell, and glucose will transport it. Again, once this both moved, this again restored to its normal position. Again, more molecule will come and it will keep on moving this glucose inside the cell against the concentration gradient. Though inside the cell glucose concentration is high as compared to outside, still glucose will be absorbed inside the cell. Now, where is energy utilized? सेकेंडरी एक्टिव ट्रांसपोर्ट डजंट यूटिलाइज द एनर्जी. यहाँ पे कहाँ पे एटीपी यूज कर रहे हैं मोमिन? सॉरी, क्या मेमन? Where we are using ATP here?
Yes, sir.
Do we use ATP? Did I mention a single word about ATP?
No, sir. But
Sir,
they both uses
ATP
pump them.
uses in sodium-potassium pump.
Here it is not utilized. Na? There is no direct utilization of ATP. That is the main difference between primary active and secondary active transport. Understand?
Okay, sir.
Yes, sir.
Now, where is energy utilized? Like you said, now sodium concentration inside the cell increase. And as I told, when your sodium concentration inside the cell increase, it will activate sodium-potassium ATPase. And now this sodium will move outside the cell and where the energy is utilized. Okay, so they utilizes the energy indirectly, and that's why name is given secondary active transport. Okay, or we can say that they utilize the potential energy of the sodium concentration gradient. So whenever the sodium concentration is very much high here outside and very low inside, and that creates the concentration gradient, and this concentration gradient is also called as a potential energy. If you remember in 12th, kinetic energy and potential energy. So this is one of the example of potential energy, and that potential energy will be utilized indirectly by the co-transport. Understand? Now opposite will be the counter transport, or also called as a antiport. Okay, it is exchanged with the sodium. So now with the same diagram we can explain. Okay, इसमें नहीं दिखाता, इधर दिखाता हूँ. Here consider this is cell membrane and this is a counter transport or antiport. Now this is extracellular fluid and this is intracellular fluid. Now here the sodium binding site will be here, and here another chemical. So consider now we are talking about the calcium. So here is a calcium binding site. So whenever
Sir, in this both molecules move in same direction.
no, it is in opposite direction. That's why name is given counter transport. In same direction is co-transport. Okay.
Okay, sir.
Another example of co-transport is instead of glucose, we can utilize amino acids. So amino acid will also can be transported. Instead of glucose, we can use chloride ion. Instead of glucose, we can use iodine. So these are the few examples of co-transport. Now this is the counter transport. Understand?
Yes, sir.
Now what happens? Sodium ion.
Sir?
Yes.
So anti-transport and co-transport difference, sir? What difference?
First, don't get confused with the anti and all. Okay. The two terms co-transport and counter-transport, or another two terms symport or antiport. Symport and co-transport are same. Counter-transport and antiport are same.
Okay.
Counter transport
Co-transport and
exists in
antiport में
antibody.
confuse मत हो. Why I mentioned this both name? Because some book has given antiport, some book has given counter-transport, some book has given symport, some book has given and
Co-transport.
don't confuse sodium potassium is pump primary active transport. These both component are move against the concentration gradient. Okay. In co-transport and counter-transport, one component moves along the concentration gradient, while another component moves against the concentration gradient. समझ में आ रहा difference?
Yes, sir.
Symport, antiport,
Yes, sir.
counter-transport or co-transport, these words are utilized only for secondary active transport. It is not used for primary active transport. So sodium potassium transport is not antiport. Okay? Okay, Shubham?
Yes, sir.
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Now, what will happen? Now, sodium ion attach outside the cell. That will increase the affinity for the calcium ion inside the cell, and calcium ion will attach inside the cell. When these both will attach, it will create a drag force of a high concentration of the sodium outside and low concentration inside, and sodium will move inside. That drag will causes again that seesaw moment of the pump, and this will move outside. Moment comes inside, sodium loses its affinity, and as the sodium loses, calcium also loses, and both are released into respectively into intracellular fluid and extracellular fluid. Similarly, sodium hydrogen counter transport are seen in kidney, where the sodium moves inside the cell while hydrogen moves into the lumen of the or renal tubules, and this is how it will causes acidification of the transport. There are other examples like sodium potassium counter transport and all those are there. So this sodium potassium counter transport which is given when the moment is occur of sodium along the concentration gradient with potassium against the concentration gradient. This type of the mechanism when occur, there we call it as a counter transport. Okay.
Yes, sir.
So then these are the mechanism of
Yes,
counter
sir.
transport, and here also energy is not utilized directly. There another term many books has been given uniport. So uniport is not any transport mechanism. So I don't describe that because it has been not given any standard book anything about the uniport. Class 12 भूल जाओ. In class 12 NCERT it is a part of facilitated. वही बता रहा हूँ आपके इस नए बुक्स में भी बहुत जगह पे symport antiport has been given in facilitated diffusion. So some
Sir,
will
in
exam
uniport,
लेके जाए.
molecules move in only one direction.
What happened?
Sir, in uniport, molecules only
Yeah,
move
that
in
has
one
been given
direction.
in report, but where is the force? Force for the moment. If it is along the concentration gradient, then it is a part of facilitated diffusion. And if energy has been utilized
Yes,
directly
sir.
from the ATP, then it is a part of primary transport. Once you pass MD physiology, we can discuss about this very much.
Yes, sir.
Okay.
Yes, sir.
So this is about our secondary active transport.
Sir,
Any
sir,
questions? Yes.
sir, sodium potassium pump and sodium potassium counter transport system both are different.
Both are different. In sodium potassium pump, the ATP is directly utilized, and that pumps the sodium and potassium along the here. ATP is not directly utilized. The moment is going along the concentration gradient. This no book has been described. Just name mentioned. That's why I mentioned this name. Detailed mechanism of this other counter transport has been not yet described in detail. Okay.
Okay, sir.
Any other question? Any other doubt? I'm opening the chat box. Okay, fine.
Sir, in kidney, renin helps in counter-attack mechanism.
That is different. Counter current mechanism.
Okay, sir.
But renin doesn't help in counter current mechanism. Okay.
Yes, sir.
Now come to the other transport mechanism. So this is a vesicular or membrane transport, vesicular transport mechanism, and this has a endocytosis, exocytosis, and some book has mentioned transcytosis. And this is for the macromolecules like large proteins or fragment of the cells, or sometimes microorganism, bacteria, and viruses also transported by this mechanism. Yeah, very good. हाँ,
आपके लैपटॉप में
स्क्रीन शॉट करना पड़ता है.
अच्छा एक मिनट
Okay, now come to the endocytosis. This is this endocytosis is also called as a cell eating or drinking, where the part moves inside the cell. Large molecules are moves inside the cell. And these endocytosis are divided into two different parts: pinocytosis and phagocytosis. Technically, or mechanism like this, both are almost same. The only difference is pinocytosis when the substance which engulfed by the cell is in a soluble form. So when a soluble substance is engulfed by the cell, then we call it as a cell drinking or pinocytosis. Means smaller substances are when they taken by the cell. That time we call it as a endocytosis. And phagocytosis when solid particle or non-soluble particles when they are engulfed, that time we call it as a phagocytosis. Phagocytosis is cell eating, while pinocytosis is cell drinking. Now, how this mechanism occurs? So, whenever uh, this mechanism occur, what happens whenever any large molecule will come in close contact with the cell, and cell want to engulf it or there by any mechanism, like receptors usually is there, and the receptors will attach to that particular molecule, and when it initiate the phagocytosis or pinocytosis, what will happen? The cell where membrane where they attach, this membrane moves inside the cell, so invaginate within the cell. At the same time, the nearby cell membrane, which is little bit away from the site where the molecule attach, will move upward movement, finger-like projection will be there, or pseudopod-like projection will be there, and that will try to cover it, and they will completely, these both pseudopods will completely engulf these chemicals, and they will come in close contact and touch each other. So they will come here and touch each other, and this portion will fuse, and this portion will fuse, and this will be sent inside the cell as a vesicle. This vesicle is many times called as a phagosomes. Phagocytosis के वजह से जो vesicle होता है, that's why it is called phagosome. So pinocytosis you will see usually during reabsorption of reabsorption occurring when by the renal tubular cells. That time the pinocytosis will be there. While phagocytosis is usually seen whenever there is a bacteria or any dead tissue or any microorganism, or sometimes large proteins are there, will come and they will attach to it, and then it get engulfed. So the three mechanisms are there, which first is attachment, then engulfment, and then vesicle formation. Okay, once this phagosomes forms, these phagosomes meet with the another vesicles called lysosomes and form phagolysosomes, and this will try to digest all these products. And once this product will digest it, if this product will completely digest, then these vesicles will dissolve into the cytoplasm, and digested products will be released into the cytoplasm. If they partially digest, then this second mechanism will there where the exocytosis, and cell will move outside the body by the exocytosis. So next slide में हम देखेंगे. Third mechanism which we call receptor mediated, but this is both pinocytosis and phagocytosis. The many mechanisms are usually receptor mediated. Many mechanisms usually occur through these antigen antibody complex mechanism or MSC molecule mechanism, and where the substance attached to each other, and that will initiate the movement of the particle. But these are specifically maintained for the chemicals like absorption of the iron or absorption of the cholesterol, which occur through this receptor mechanism, and for that we use this receptor mediated endocytosis. So this phagocytosis, which cell usually does? Not all cells could able to do this phagocytosis very effectively. There are only few cells which present in our body which causes this phagocytosis, and those are immune cells. One is macrophage, which present throughout our body. They are like a patrolling police, and they'll keep on moving in our body, and whenever they'll find any dangerous substance or organism or anything, they will try to engulf the phagocytose it and try to kill those bodies. Another is neutrophil, is present within the blood, and whenever this infection occurs or an injury occurs to tissue, it will move outside the blood and can cause microorganism or dead tissue phagocytosis. Third is activated monocytes. These are nothing but the blood counterpart of the macrophage. So the activated monocytes are much larger, and like a neutrophil, manifold they can cause phagocytosis more than a neutrophil. Neutrophil is very small, but they're fast to active, while monocytes are slow to active, but they're very effective, and then they also can cause phagocytosis. So many times phagocytosis also act through the antigen antibody complex formation, and that can initiate or encourage the phagocytosis. Then another chemical complement system are also present that can also initiate the phagocytosis. Then various other chemicals which can attach to the cell, and then can also increases the phagocytosis. Then come to the exocytosis. it is just opposite of the phagocytosis, where the particles is expelled out of the cell. And in this, if we see the cell membrane wear and tear is continuously going on. So without breaching the cells, just it expel out the particles. So just consider the phagocytic cell digest the bacteria, and that bacteria is not completely digested. Some portion will remain inside the phagolysosomes. Now these phagolysosomes will move towards the cell and cell membrane, and they fuse its membrane with the cell membrane, and it forms a continuous cell. The continuation of the cell will be there, and the particle will be moved outside. Similar way, the various hormones are released by the exocytosis. Even neurotransmitters are also released by the exocytosis. And even enzyme released by the various GI tracts, that also done by this exocytosis. So when the exocytosis occurs for release of the various substances, this exocytosis does occur, and this usually utilizes the calcium. So whenever there is increased calcium concentration inside the cell, it can help for the release of the exocytosis. But the phagocytic cell which can cause the exocytosis, neutrophil doesn't cause exocytosis. It only causes phagocytosis, while monocytes can cause phagocytosis also and exocytosis. That's why monocytes can have a very long lifespan and can have thousands of the cells they can phagocytose and kill the thousands of the bacteria, while neutrophil has a limited 100 to 200 cells only it can phagocytose, and up to that time it will start dying because waste material will be keep on adding within the cell. Apart from these functions, the various organelles will become old and disintegrated. That organelles also can digest within the cell and reabsorb, or if it is not possible, then they can be expelled out through this mechanism. This is how the transport mechanism works. Then come to transcytosis. It occurs when one side is phagocytosis, another side is endocytosis, and exocytosis continuously occurring without making any change in that chemical. Then we call it as a vesicular transport within the cell. It is also called as a cytopempsis. Again, this is usually seen in renal tubules where the vesicular transport will be there. And there similarly, first phagocytosis, vesicle formation, transportation, and then docking of the cell are called exocytosis. These three basic steps are involved. And very less books has been described more about the transcytosis. Then there is a parasitosis where it occurs through these junctions. It is not within the cell, but it is in between the cells. From one side of the lumen, it moves to the another side of the lumen. This is called parasitosis, where the chemical has been transmitted from one side of the cell to the other side of the cell. So that's all about the transport mechanism. Now what you want me to take? Either I start with a new topic, MCQs.
MCQ, sir.
MCQ.
The last 15 minutes should be MCQ only.
Okay. So we'll start with a new topic.
You can start with the summary of the new topics.
Okay.
Sir, I have a doubt.
Yes.
Clathrin mediated endocytosis is similar to receptor mediated endocytosis.
Yeah.
Okay, sir. Excuse me, sir.
Yes.
Sir, how a cell can identify whether it has to be apoptosis performed or this endocytosis, exocytosis has to be performed to the organelle?
फिर से एक बार सवाल पूछो, आराम से हिंदी में भी पूछ सकते हो.
Sir, how can a cell membrane identify that it has to be apoptosis performing or endocytosis or exocytosis has to be performed for the worn out organelles and all?
Sorry, I didn't get your question. See, apoptosis is different. Phagocytosis is different. We usually compare apoptosis with necrosis. Okay, and phagocytosis occur with both cells, whether it is occur apoptosis or whether it occur necrosis. In both conditions, it gets phagocytosed by the macrophage.
Okay, sir.
There one question, actually, what I thought you're asking, phagocytosis, suppose a human cell, and how they can decide which cell to be phagocytosed or which cell to be killed and which cell should not be killed.
Yes.
Actually, this was my question, sir. I was not able to properly ask.
इसलिए मैंने बोला था आप हिंदी में भी पूछ सकते हो. तो many times the phagocytic cell, uh, which cell is supposed to be killed and which cell is supposed to or not to be killed. It is like a military. These phagocytic cells are like a military persons. Now military persons, they get a target कि फला यूनिफॉर्म वाले को मारना है, फला यूनिफॉर्म वाले को नहीं मारना है. In same way, this phagocytosis cell works. So whenever our immune cell work, they work on MHC molecules. So major histocompatibility molecules. They present on almost each cell of our body. And whenever a cell which come with a different type of the MSC complex, and these MSC complex are unique for individual, except it is a identical twins. Identical twins nahi hai, so with each individual, this MSC complex is unique for that each individuals. Okay, so whenever that different MSC complex will come, then this target cell will identify those MSC molecules and try to kill those cells. This is how they will identify the foreign bodies. Understand?
Yes, sir.
Okay. Second thing, there are like military जैसे होते हैं वैसे सामने वाले भी होता है, उसको भी पता है कि सामने military है मुझे मारेगी, so they can disguise these militaries. So there are many viruses in we got infected. Those viruses causes down regulation of the MSC molecules. On our own cells, they started down regulation of the MSC molecules so that the immune cell should not act on it. So there is a separate mechanism for that. Another immune cell is there, which we call it as a natural killer cells. And these natural killer cells has only one target. They are like a black cat commandos. जिनके पास flag है उनको छोड़ दो, जिनके पास flag नहीं उनको मार दो. So moment a cell which contain MSC molecules, this natural killer cell will not act on it. Moment they will find any cells without MSC molecules, they will try to kill those cells directly. That's why they are called natural killer cells. They need the flag, don't kill me. Like that flag should be present on there. If they don't say anything like that flag, they will start killing it. If there is a presence of the MSC molecule, they will not act. Moment there is absence of the MSC molecule, they will act on it. Any other question?
Sir, autoimmune disease is related to this type of function.
Yeah, you can call like
Autoimmune
that.
disorder.
Natural killer cells also kill cancer cells also, no sir?
Yeah, yeah. फिर कैंसर सेल में भी एमएससी मॉलिक्यूल्स का रिप्रेजेंटेशन कम हो जाता है.
Okay, sir.
Any other question?
The student is asking what type of questions come in
Yeah.
physiology.
Okay. अभी पैटर्न चेंज होगा. Previously it was just simple MCQs used to come and you have to write. There are many one-liner MCQs. And then there are many little bit complex MCQs. But this time what I heard is we'll get a conceptual based MCQs, like more of the clinical cases. So application of this, what knowledge you are getting, application of this knowledge will be there. Okay.
Sir,
Yes.
the questions given on the Prosium site are enough for us.
Okay, this is not NEET exam. You are going to be doctor. Questions will never be enough. Understand?
So, sir, is there any specific book for MCQs?
I don't advise to read a specific book for MCQs. You first read about the textbook, then before exam you can read about the MCQs.
Sir,
Okay, sir.
सर
Yeah, yes.
और सर प्रैक्टिकल मतलब किस तरह से हो गया? प्रैक्टिकल होने वाला है फिजियोलॉजी में?
होने तो चाहिए. मैंने एक स्कीमेटिक मैकेनिज्म बताया है. लेट्स सी हाउ इट वर्क्स. अभी तक जो प्रैक्टिकल होता है, the practicals are on module basis are there. Once you come here, you will have regular practicals on module basis. But in addition to that, we want more practical for you. So let's see how it works. Okay, in these practicals, you will learn how to examine the patients, how to examine blood or urine sample. All those practicals will be taught. Urine sample का ज़्यादा detail आपको biochemistry में पढ़ाया जाएगा. But in physiology, more of the blood and clinical examination of the patient.
Excuse me, sir.
Yes.
Sir, apart from your classes, sir, कौन सी textbooks refer करनी है sir?
इंदु खुराना, A.K. Jain, Guyton.
Thank you, sir.
Sir,
Yes.
Sir, please explain transcystosis once again.
Transcytosis, it is a combination of endocytosis and exocytosis, but the chemical structure of the substance which is transported through the cell will not change. Okay, it will remain same. तो इधर albumin आया तो इधर भी albumin ही आएगा. That is the difference between. अब तो सब endocytosis exocytosis हो जाएगा तो हो गया. But in this, the change doesn't occur. Same substance from one side will goes in the same form to the other side. So one portion it will cause endocytosis, and then another portion will comes and it will causes exocytosis. This is how the substance will move from one side of the cell to the other side of the cell. It is not inside the cell. It is moving from one side of the cell to the other side because here is a tight junction. It cannot move through this tight junction.
Okay, sir. Thank you.
Sir,
Sir, in this, what is caveole?
ये vesicles को बोला है क्या ये?
Okay, sir.
I took this diagram from internet. I couldn't find in books.
sir, APC book is good or not for physiology?
Sir, phagocytics or NK cells, these all mechanisms will be depends on receptor cells, sir. Receptors.
Please, आप सब mic वाले जो बोल रहे हैं, but there are many people who are writing. So one has asked me, docking. What is meant by docking? Docking उसी तरह से जैसे शिप जाके पोर्ट पे रुक जाती है, पार्किंग करती है. सेम वे दे आस्क डॉकिंग इन द सेल. तो अल्ब्यूमिन आ जाता है और सेल phagocytosis होता है, and then it goes and park in opposite side of the cell. Exocytosis को they are use the word docking in the cell. So it just parked there without making any changes. Then another one is asked about proton pump. So proton pump. Okay, proton pump हमने इसमें पढ़ा था. Sodium potassium pump. Okay. So proton pump is a potassium hydrogen pump. Okay, here potassium is a just accessory. So since potassium is not important, moment of the hydrogen is more important. So many times it is called as a hydrogen pump, and hydrogen plus also named as a proton. That's why name is given proton pump. These pumps are present in parietal cells of the stomach or gastric mucosa. Parietal cell of the gastric mucosa. There it present on the canaliculi, and it will push the hydrogen ion much than timer concentration in the canalicula as compared to cytoplasm. So hydrogen ion will move against the concentration gradient into the canalicula by using direct ATP. And this is why it is called proton pump or potassium hydrogen pump or sometimes hydrogen pump.
Sir, APC book is good for physiology.
Which?
APC.
I don't know about it.
Sir, sodium potassium pump is a example of voltage gated pump.
No, voltage gated pump नहीं होता है. Voltage gated channels होता है, and they are example of facilitated diffusion. ट्रांससाइटोसिस का ही अदर नेम है साइटोएम्पेसिस. ओके, एकेजन बोल रहे. एकेजन इज गुड बुक. Okay, you are talking. I thought you were talking about book. Antigen presenting cells are present in the cells. When we are talking about hematology, we'll talk about this in detail about antigen presenting cells. These are the cells which phagocytose the foreign cells and take the splits of the different antigens, and those antigens are present on their surface along with the MSC complexes. These antigen producing cell or antibody producing cells like lymphocyte B and lymphocyte T, they cannot identify antigen abruptly. They need antigen to be presented, or in the plate they should be served along with the MSC complex. Then only they can identify. And that's why these phagocytic cells like monocytes will act as a phagocytes, and they present this antigen to the T cell and B cells, and then T cell B cell will find that antigen whether it should be act or not, and if they act, they'll produce particular antibodies or particular chemical. Like B cell will produce antibodies, and the antibodies will bind with the antigen, and we'll have different mechanism to deal with those antigens. Or when T cell will be there, they'll bind with the antigens and start puncturing the hole and try to kill the cell. Samolingam is not a good book. अब तुमने खरीदा इसलिए मैं उसको अच्छा नहीं बोल सकता ना.
सर कोई रीजन
Autophagy,
वगैरह तो होगा
when the
ना.
cell will try to kill itself and try phagocytose will be there. Notes आप खुद बनाओ. You are expert in the making notes. Okay. अब कंसल्टेंसी नहीं दिया करके उसकी तारीफ करूं मैं. Guyton, very nice book. It's one of the best book. GK Pal is also very good book.
So when will semester exam?
Sorry.
So when will semester exam occur?
Most probably February. Good. Use Guyton exam. Anything else?
So apart from exam, are there weekly tests and all tests?
If you want, I can arrange a test for you. But I need approval at least from 200, 300 students.
Yes, sir.
So
Okay, panelist, can you demote?
Will approve.
Panelist, can you demote yourself to the as attendee on your own?
But
Okay, sir, we
college
can
is
do.
not started, so Sir, college is not started, and so how
Sir,
exams
you
will be
only do, sir.
conducted?
Yes.
We cannot
Sir,
demote ourselves, sir. You can do.
You can leave
Sir,
the
they are
webinar and
only.
join, then you will become a normal student.
Sir, there are only 36 panelists, so maybe we can just give you our answers.
Okay,
Anyways.
fine.
sir, college is not yet started, so how exams will be conducted in February means?
Sorry?
Sir, college is not yet started, so how exams will be conducted in February means we are late in our session, so.
एक मिनट मुझे लिखने दो.
Sir is sending a question about taking test.
ये चल रही है. पोल डाला है, प्लीज गो थ्रू इट. डोंट वरी मच अबाउट द एग्जाम. आप लोगों ने भी एमबीबीएस लिया हुआ है. हर you have to give at least 60 to 80 exams every year.
We can't vote,
Don't
sir.
worry.
We will
Huh?
tell you orally.
Sir, we are not.
We will tell you orally.
Don't worry, the panelists cannot vote. Let the students vote.
Sir, I was asking that college is not yet started, so how exams will be conducted
College
in
has been
February?
started here. Right now, college has been started. Classes start हो
But
गए
sir,
मतलब
not
college start.
okay, sir.
The students are
Gaurav,
there.
everyone can't speak on the mic. Yes, Sanaya, it is your first lecture. I can send you link of other 3 lectures. You can go through it. Please don't ask these nonsense questions. Kazma se kaun hai, yahan se kaun hai, wahan se kaun hai. You can go to the WhatsApp group and talk all those things. रिद्धिमा, it is both MCA classes and college classes. दोनों के अलग अलग फिजियोलॉजी नहीं, सबकी फिजियोलॉजी एक जैसी है. Yeah, I already told you about my number. You can WhatsApp me. I will send you link. Okay, please stop sending messages now. Just let my number display. Okay, everyone got my number?
Yes, sir.
Okay.
Yes, sir. Yes, sir.
Sir, panelist cannot participate in the poll.
Yeah, I know. So poll time, I will keep it for 1 more minute and then I will end it. No idea. Offline classes, I don't have any idea.
Sir, it will be in MCQs or description.
No idea about NMC brochures.
Sir, exam will be conducted in MCQs or descriptive wise.
Most probably MCQ में ही आएगा आपका.
Okay, sir.
Okay, so this is your poll option. So can you see result?
Yes, sir. It is a good result.
So okay, I will plan accordingly and I will let you know. Okay. And if you want, we can discuss also about this MCQs after exam. Okay, then I think we should stop here. Next class, we'll talk about membrane potentials. So please read membrane potential and then come. After that, I'm jumping from 1.5 competency to direct 1.8. Please don't talk. Please don't talk. So next class I will talk about membrane potentials. So if you go by syllabus wise, competency wise, it will come under 1.8 and not 1.6. Okay. Just I will show you our syllabus. So right now we are talking about general physiology. In general physiology, those who are new, please you also concentrate. We are already finished the 1.1, that is mammalian cell physiology. In 1.2 about homeostasis, 1.3 about intracellular communications, 1.4 apoptosis, and today we finish 1.5. Next is supposed to be 1.6, that is fluid compartment and all those things, and pH and buffer regulation. But this will take later. First, we'll describe about the membrane potential, because membrane potential are more linked with the transport across the membrane. So we'll talk about the membrane potential, that is resting membrane potential, action potential, and local potentials. And then we'll start with the body fluid and pH and buffer mechanism. This 1.9 is a part of practical, so we are not going to complete this in a theory. Okay? Okay, bye. Have a nice day. If you have any doubt, any questions, you can ask me in a Telegram group. Okay. And I got too much messages. I will reply with a link of Telegram group. And if you had your friend who already in Telegram group, please share those link with your friends. Okay, 1 minute. जिसको Telegram group की link चाहिए, please. Okay, someone has already sent. So you can leave. Those who want to leave. And after 5 minutes, I will switch off this class. Okay, join Telegram group. In that, I will send the link.